Really kind of honored to be the final speaker in this first session to really talk about kind of a twofold approach of our biofabrication. So it's beyond just printing because I'll kind of go into that and on the International Space Station. And the tie into food actually began when this whole project began. So this came out of a kind of an internal company development in about 2014. We approached NASA for the first time in 2015. My background is regenerative medicine, wound healing, and cardiovascular transplants. And we looked at it from, let's look at ways to address tissue shortage, address organ shortage. But the conversation kept coming back to food from NASA. So, NASA kept saying, it's like, well, it's great to produce organs, but what can you do about food? And well, that's really cool for tissue, but what about food? And, food is one of those things that we've been thinking about internally. We have other partnerships that are in the stages of development in cellular agriculture on the ground, both for meat production, for plant production, since Techshot owns and operates 7 pieces of hardware on orbit right now, and then manages 2 others, everything from advanced greenhouses through research furnaces, and then all of our work, the company's 31-year history, really in bioreactor development and life sciences dominantly. So quick shout out to Jessica Meir and all the great astronauts on the International Space Station with whom none of our projects could happen. More than a great set of hands. It's a great set of hands and a brain behind it. The troubleshooting is always ongoing. And to answer one question that was in the thread earlier, you don't necessarily need to have a cell culture expert in space to do this work, but having smart people really makes the job easier. So let's start off with the, why print? Because, isn't culturing hard enough all by itself? Well, what kept coming back to us and coming back with NASA and with our other conversations is all the psychological benefit of a mouthfeel that you can improve with a printed meat product. And going beyond food is just a nutrient delivery system. So, we routinely hear this from colleagues that are astronauts coming back on everything is bland. They're looking for fresh food. They're looking for a variety of food. Scott did a great job of kind of setting that plate with his sashimi to make me hungry since I'm missing lunch. And kind of going through this whole thing, it's the diversity and the freshness that everyone is after. And people aren't going to settle for less when we start sending civilians and individuals to space beyond professional astronauts who, go for the good of the country, go for the good of the science. Pretty soon people are going to go for the good of their own wallet or go for their own adventure, and they're going to want a better quality of life in space. So we really need to address this food issue, as well as improving the appearance, looking for the satisfaction, as well as visual cues for satiation. So, people will eat less if they're eating something that looks good. And then the links to memories and familiarity. So, as Scott mentioned, a short mission to Mars is 3 years. That's a long time to be homesick. And there's very distinct jobs that have to happen for crews to exist in outer space on a foreign planet for 3-year time periods. And if you start to become homesick, start to become depressed or stressed, you lose your efficacy as a crew member. And at least in the early crews, we won't have enough redundancy in those crews that someone can, take a week off because they're feeling homesick. So these are all important aspects, and these all tie back to food. So for us, I'm just kind of jumping backwards to bioprinting and what it really means for us and with our bioprinter. So our bioprinter is just really a collection of sophisticated dispensers, motion controllers, temperature controllers, imaging system, and software to reproduce a solid material out of various other, a solid material out of various materials and bioinks. And for our biofabrication facility, or our BFF, hopefully someday to be your BFF, we use 4 print heads to print our biological constructs. It gives us high dispensing precision with dispensing tips between about 50 and 150 microns. So roughly the size of a human hair. So you can understand for placement, whether it be for texture or for nutrients or for cells, that we can put things almost exactly where we want them. And then we can look at what those bioinks really are. So again, this program for us developed out of a need for or desire to address the organ shortage, but an organ is simply the same thing as a food, whether your source is going to be a human tissue for transplant or a animal tissue for consumption. You're really looking at a bioink that's comprised of proteins and nutrients and other things that will affect the cells so they can continue to grow, as well as provide the cues for them to mature into what you're targeting. Our cell work is being done almost exclusively with induced pluripotent stem cells, so we can allow multi-cell lineages from a single cell source. Talk a little bit more about this in a second, but really that IPS process is going to unlock a lot of keys to how to move this forward and how to make this more effective. And then the bioreactor. So that really induces chemical, mechanical, electrical signals that can transform a printed construct into a functional tissue. So internally, we like to use an expression, when is a pancake a pancake? So pancake batter is essentially what comes out of your bioprinter. And nobody is really satisfied with a pool of pancake batter on their plate. So it has to be heated, it has to be cooked, it has to be flipped, it has to be monitored, and then the final product everyone enjoys is the regular pancake. That's where the bioreactor comes in. So, we're well beyond just a way of dispensing material, but now a way of taking those materials and get them to interact into form the solid or form the tissue or form the product that you really want to consume in the end. So the overview of our facility, again, it's a 3D printer, which also has culturing conditions to form functional tissues. But why microgravity? Why did this whole thing start in the first place? Well, the advantage of microgravity is there are many things you can do in the absence of convection, in the absence of buoyancy. So you can use lower viscosity inks, you can put nutrients where you want them, and print biology for the sake of biology. This becomes really important when you're looking at a food product because it's all that extra filler, all those extra materials that you need to add structure as you're printing that really detract from the nutrient value of what your end product is going to be. I'll get to what BFF looks like on the next slide. It's an L-shaped triple locker for those that speak NASA, gantry style printer based on the NScript 3DN series bio assembly tool. So again, NScript has been a great partner for Techshot in this, and we're really looking on multiple fronts for austere locations for bioprinting and adding this capability for printing food, printing tissue, printing other products. And whether that austere location is Appalachia, which is near and dear to our heart here in Southern Indiana and Kentucky, whether that be Sub-Saharan Africa, whether it be India, or whether it be the moon, all austere locations, all oftentimes with limited technical resources for these high quality, high-end products. And we're doing a lot of work with remote sensing, with remote control, and with artificial intelligence for automation, really to enable these systems to operate autonomously or near autonomously. So here's what BFF looks like. And to put this in perspective, that print chamber on the left of the screen is about the size of we'll say a dorm refrigerator. That's form factor is 2 express rack units, which would mean it's roughly a 2 foot by 2 foot by 2 foot cube. The electronics compartment on the lower right, that forms the L. And then our tissue culture system, which is by name our ADSEP, it's our advanced space processing and experiment processing platform, legacy hardware back from our shuttle era that we've updated to work on the International Space Station and beyond. Inside, this is kind of what the platform looks like, where you can see the color-coded 4 print heads to try to make it easy for crew members adding and removing inks. So all you have to do is match colors and dispense it the way you need to. On the right-hand picture is looking at it with 2 dispenser brackets mounted to the smart pumps and our tissue processing cassette. So the cassette is eventually what's going to be the bioreactor, and we print directly into that bioreactor and then transfer it into an incubator that has all the formal controls to, again, to condition that tissue into a final product. The bioreactor itself is a self-contained system which provides feed media waste as well as an imaging system, and then the reactor that can apply chemical, mechanical, and electrical stimulation. This kind of touches on an area that's that Scott brought up, and understanding that waste stream, because the way that we typically do cell culture on the ground is it's single pass, that you feed a cell culture media, you collect the waste, then you start from scratch with virgin media. As you scale that, virgin media is going to become, too expensive. It's going to become too bulky, and it's going to become too hard to produce. Much like the way the body produces it and uses things right now. You don't need an orange just for the vitamin C. There's many other products, and just because you can deplete one aspect of that media doesn't mean you need to replace the whole media. And we need to add a lot of new sensor technologies to really monitor what the growing tissues are needing and what the growing tissues are using from their media, so we can replace only what's required to really limit our use of water, to limit our use of other resources. So our print volume right now is we're printing in a 31 by 23 by 23 millimeter block. While that sounds large in the bioprinting aspect, on the food size, that's roughly only 1/7 of a single food portion of a 4-ounce, 100-gram meal. And really what that allows us to do is apply everything and use this as a test bed to understand what's going to be necessary for scale, both for, again, for our company's primary mission of tissue for transplant, as well as our secondary mission of supplying food for low Earth orbit and beyond. But again, just understanding the complication of this, that our best bet right now is still only 1/7 the size of a single portion of a single meal for a single astronaut, just to kind of understand the scale of what we're looking at. So the other piece that I touched on briefly in the beginning was our iPS cell technology. We call it a cell factory. It's a reprogramming facility to transform mammalian cells into induced pluripotent stem cells. We're using a non-viral reprogramming technique that luckily everyone is becoming much more familiar with because of the Pfizer and Moderna vaccines, because it is an mRNA-based system as well, which produces a stable transformation. So what we can do is have a near unlimited cell source. The advantage of the pluripotent stem cells is they will continue to multiply. And as Hilda mentioned, and thanks for the plug earlier, again, a great partner in the ISS world, that cells maintain their stemness in microgravity, which means that while on the ground, when you produce an iPS cell, you have to constantly keep low densities and special conditions to prevent those cells from differentiating into a different cell type. On orbit, it seems to be less an issue, that these cells can grow to higher densities, can grow to higher confluence, and still maintain their stem nature. And so we can use those for producing the other tissues we need. We have the proven trilineage capability of those cells, so we can move into any printed tissue that we want or any printed tissue that we need. And mostly because, again, tissues aren't single cell types, we're going to need to add multiple components to a single end product, and then either differentiate them before printing or print a progenitor cell, so not quite fully differentiated cell, and use those and allow them to multiply after the printing process during the maturation process. Again, we're looking at growth densities right now of about 100,000 cells per square centimeter for the progenitors, and up to 500,000 cells for the differentiated, which sounds like a lot up until the point that you think that we can look at our target tissue, roughly 30 million cells per cubic centimeter. And if you really start expanding that out to a single 100 gram or 92 cubic centimeter tissue, you start to realize that the cell numbers themselves become a really daunting enterprise. For our printing process, we can usually print between 1 and 5 million cells per centimeter, which then requires 3 to 5 cell doublings from the time that we print to the time that we have a mature tissue. And again, those cell doublings take time. Some cells will double in hours to days, and some days to weeks, depending on how differentiated the cell is and what the cell lineage is. And all of those will need tissue-specific media containing cell-optimized basal media, so just the base food, as well as defined growth factors and supplements. I've been looking at the chat channel. There's a lot of great people that are working in this field, and we're going to have to leverage all that to understand how to move forward, combining that technology with advanced sensor technology, so we really understand what we need and when we need it. So looking at printing, the other aspect of what we're doing is we're adding some sacrificial inks. So this is what sacrificial ink printing looks like on the ground. So we can take these, span it over a 25 millimeter gap, and you can see in a short period of time, those bars tend to drop down, tend to settle, even the sidewalls are settling in. This is the hazard of bioprinting on the ground, is that gravity really is a huge influence and really can impact the ability of positional control with low viscosity materials. In image C, we're looking at a low viscosity print and gel formation to understand what's going on with tissues on the ground. Our ground testing, we're looking at printing different cells in different areas with different materials. So if you can look at this either for blood vessels moving through a tissue or for fat bands through sashimi grade tuna, these are both very similar aspects of putting high spatial resolution in different regions and getting them to grow into a solid material. You can see from image D there, those are vascular cells. It's a fairly good live to dead ratio, although you start to see as it moves deeper in the tissue with the top of the tissue near the top of that image and then down into that tissue, roughly about 300 to 400 microns, you start to see a lot of red showing up. Red is dead. It's obviously not what we want. We need healthy live tissue. So perfusion, again, for thick tissues, whether it be for food or for transplant, is still a huge issue to overcome in this field. Our flight experience, we're looking at, this is from the SpaceX 19 mission. So this was the fall of 2019, before we lost a year. And what we're looking at is kind of reproducing those print images and those print tissues as we start growing some of these tissues in microgravity and understand how our bioinks are related, how the scaffolds are related. And what you can see on the right-hand image is you can start to see a weird meniscus effect where we actually have a convex meniscus instead of a concave meniscus. This is driven by surface tension rather than gravity that you would see on the ground. And this really changes the way that we have to look at our materials and start to look at the interaction of cells and materials and where we place them. It's a technique called mechanical pinning, which is necessary for moving fluids around in microgravity because fluids aren't going to stay where you want them simply because of sedimentation or gravity, like on the ground, that you have to add physical constraints or electrical constraints to move fluids to where you want them. But we were actually able to take all that into consideration and print tissue within our reactor. What you can see is that's after filling it. And in the right-hand image, you can see another massive challenge for doing cellular agriculture, especially culture-based agriculture in microgravity, is the bubbles go where they want to go. Again, driven by surface tension and driven by other factors, but not buoyancy. So those bubbles will stay wherever, throughout the media. So adding sophisticated bubble traps, adding sophisticated means for pinning fluid for filling, all become serious issues for transitioning what we know about cell culture on the ground to cell culture in space. And finally, we start to look at some of our early lessons learned on moving these future planned missions, both to optimize the delivery of nutrients and stimulation on subsequent missions, as well as looking at the chamber design. Again, like I was speaking, the mechanical pinning to change the potential material interactions, the shape, and even potentially electric fields, both to move fluids around as well as to get rid of trapped air. While air may seem like a good idea to be trapped in the fluid, if it comes in contact with the growing tissue, it tends to cause necrosis or cell death in that region. So we really need to study more of these surface tension driven effects to understand the various bioinks and biomaterials, as well as the shape presented within the bioreactor, and then use new designs of the bioreactor so we can get away from taking up space with sacrificial ink and start to use physical, mechanical, or electrical constraints to hold those tissues, hold those inks in place. Because again, everything that you have to add that you don't need in your finished product is wasted space, it's wasted volume, it's wasted resources. And then looking at moving and improving thermal characteristics of the cell chamber. It's difficult to test based on groundwork when you start to think that in space you only have conduction and not convection. So moving away from convective heating, unless you're using forced convection, and even then, it's an inefficient heating and cooling process. So that aspect alone becomes a challenge when looking at pivoting to orbital cell growth, orbital tissue growth, and then looking at what's really required as the population of cells and cell manufacturing in space as this whole lesson goes. That being said, it's an exciting process. We're happy to work with this. We're great to see that the work that Dane and the Methuselah Foundation is doing pairing with NASA and the Deep Space Food Challenge. We're very interested in partnerships in this approach and looking at whether it be this bioprinting platform, whether it be our greenhouse platforms, whether it be other cellular agriculture with our bioreactor technologies, really looking at a multi-prong approach because nobody wants to eat the same thing every day, and no one answer is going to win this project and help us become less dependent on roaming agriculture on the ground or in space. So with that, I'd like to say thank you and open up for questions.
Great. Thank you so much. And it was really exciting to kind of see the photos of how the 3D printers actually kind of look like in their environment and the progress there. We have a couple questions that are coming in that we'll tackle now. One is, would lack of gravity or microgravity also affect adherent cells?
Yes, absolutely. So, it works great the first time when you send them up attached, but splitting cells in microgravity is a daunting process. We found that your best bet right now is relying on the electrical charge of cell membranes and looking at seeding cells, either putting it in a low G centrifuge or using electrically adherent surfaces to force cells to drive them to the surface. But adherent cells become a large problem. And so we're looking at pivoting a lot of our work to non-adherent culture just for the ease of it.
And in terms of running experiments on the ISS, what kind of communications do you have with the astronauts or researchers? And is there the ability to make, for example, remote updates to software or anything like that?
Yeah, that's a great question. So anytime an astronaut is working with our systems, we're in direct communication with the astronaut. So on that first picture I showed with Jessica hanging upside down in front of our printer, we were on the other end of that headset that she was wearing. We also have remote commanding capability for all of our hardware systems that are on orbit. It's not a great bandwidth, but it's a pretty good bandwidth, and it's better than we're going to see on the moon, and it's certainly better than we're going to see on Mars. But like I mentioned, with the kind of AI and machine vision pieces of it, that even in microgravity, a few second delay for anyone that's experienced in 3D printing can make a big difference in your print. So adding some inherent intelligence into your system is really what's going to be required, from understanding that a few seconds to a few minutes to 20 minutes as we pivot to Mars.
We have another question regarding to just the 3D printing technology in general, which asks, what kind of texture pattern goes into 3D printing for food or the food applications that are going on in the ISS? For instance, when using a typical printer with PLA or some materials, there are lines or gridding. What is this like for food?
So, we haven't actually been involved in anything that's been printed and consumed on orbit. So we really look at, for the printing that we're doing right now for tissues, on really tweaking our bioinks based on what the cell type is that we're going to be printing, so that our vascular tissues look vascular and have the right protein mix. Our muscular tissues have the right mix for extracellular matrix. So, I have a big background in matrix biology, so a big plug to all those holdout matrix biologists out there that, our new day is coming in cellular agriculture.
Great. And I think I was maybe overexcited when I heard about the cell cultured salmon sashimi that we might be eating on the ISS. So
Yeah.
We have another question regarding radiation. It was brought up that radiation is a big problem, and NASA is doing research to address potential challenges with radiation. Does your team have to deal with any type of radiation in space that might affect the experiments?
So we're looking into it. So, ISS is fairly protected because, it's inside the protective, the protective orb around the Earth. So while radiation is higher on the ISS than it is on the ground, it's not high enough for us to have a huge impact over a 4-week culture. So, from cell expansion to printing to return, we typically run on about a 30-day mission. And in that, we're not seeing any genetic markers of damage to cells by radiation. However, moving to the moon or moving to Mars, it's absolutely going to be a bigger issue. And high energy radiation is something that we will have to take into consideration for both the protection. And the problem with radiation is that, depending on what your radiation source is, it either has to be a heavy metal or it has to be, high density polymer to protect yourself from it. So it's not a one size fits all, unless you have an unlimited water source, and then you can just surround yourself in a big bubble.
Great. And we have one last question regarding the cell factory. Is the cell factory facility fully automated, or does it need astronauts?
Right now, the system that we're developing needs astronaut time insofar as changing feed and waste bags, but everything else within the system is fully automated.
Great. Thank you very much. As any additional questions come in, feel free to put them in the Slack channel, and we will pass it along to Anita, who will close out this session block. Thank you very much, Eugene.
Thanks, Alex.
Thank you, Alex, and thank you, Eugene, for this interesting presentation.